Good evening, everybody. Thank you very much. What a fantastic audience here tonight. We're pretty much full in one of our biggest lecture theatres, so it's great to see you. And especially great to see the younger colleagues in the room. Well, a very, very warm welcome to the University of York. My name's Charlie Jeffrey. I'm the Vice-Chancellor, the head of the university, and it's a tremendous pleasure to have everybody here tonight, including our speakers, from whom we will hear soon, to celebrate the official opening of the Eleanor and Guy Dodson Building, which confusingly is not this building, but it's somewhere else. But bear with us, we're marking the opening of that building. Now, in a moment, I'm going to be handing over to our chair for the evening, Professor Caroline Descent, who's the head of the Department of Chemistry, and she will give a full introduction to our speakers tonight. We have Dr. Richard Henderson, the Nobel Prize winning scientist who did much to develop modern cryo-EM, which is what's happening in the Dodson Building. And we'll also hear from the academic lead on the cryo-EM facility here in York, Dr. Jamie Blaza. But before that, I wanted to reflect on the incredible work now taking place in the Eleanor and Guy Dodson Building, which we're celebrating today. That space enables York Structural Biology Laboratory, YSBL, and its renowned research into the molecular structure of biomolecules to move to a really new and higher level. It's an absolutely state-of-the-art facility with the extraordinarily demanding environmental conditions that are required to have equipment for structural biology operate at peak performance. I am told that per square foot is by far the most expensive building in the university because of the sensitivity that you need to do that kind of work. Now, YSBL has a fantastic history of both basic and applied research. Among its past achievements are fundamental work on the structure of insulin that ultimately led to both slow and fast-acting versions of that hormone for different medical situations. And Eleanor Dodson, here on the front row, was a key figure in that. In a different sphere, YSBL's expertise in mechanistic enzymology has helped develop the enzymes that are now used in low-temperature washing powders and other energy-efficient industrial processes. Now, those two things were among many examples highlighted in the nomination that led to the award of the Queen's Anniversary Prize to YSBL in 2019, and that is the highest national honour awarded in UK further and higher education. Looking to the future, we'll be hearing about how YSBL members now collaborate with our Centre for Novel Agricultural Products, another world-leading research centre on this campus, and that collaboration has greatly improved, for example, our understanding of how algae so efficiently take up carbon dioxide, with the hope that by understanding that process we can engineer better processes into other forms of plants to combat climate change. And I'm especially delighted that this facility is being opened as part of the university's 60th anniversary this year. 60 years ago, this university was founded on a number of interdependent and, I think, inspiring principles. Principles like these, that anyone who could benefit from a York education should be able to do so, regardless of background, gender, race, or financial circumstances. That our research should be shaped around the idea of the amelioration of human life and conditions, and that our students were to be citizens of the world. And in 2023, we've distilled these principles into a simple message: the University of York exists for public good. We believe in the transformational power of universities to bring positive impacts locally and globally. our student education, our research, and our engagement with partners within and beyond higher education, within and beyond the UK, have the potential to drive social change, technological progress, and enable innovation, productivity, and tackle global challenges. And even though at times we hear more critical views, we hold fast to the belief that because universities like ours are at heart places of exchange of ideas, views, and cultures, we play a key role in enabling our and other societies to understand and engage with each other and the world around them more effectively and harmoniously. YSBL's work must, against all of that background, surely therefore be regarded as an absolute exemplar of what our founders meant by the amelioration of human life and conditions. And I salute the generations of incredible researchers who've given so much to the pursuit of new knowledge and the application of that knowledge for public good. And speaking of those generations of researchers, I want to highlight two incredibly important people in the annals of this university's history. The building elsewhere on campus, whose ribbon was cut earlier today, is named to pay tribute to the pivotal role played by Professors Eleanor and the late Guy Dodson, who established the York Structural Biology Laboratory, YSBL. And we're truly honoured that here in the front row we have Eleanor with us tonight and in the audience with her family to join us in this official opening of the building inspired by her and Guy's crucial research. And Eleanor will be joining us as a speaker later on in the evening as we officially mark the opening of the building named in her honour and her and Guy's honour. The construction of the building was funded by a generous grant from the Wolfson Foundation and the University of York, while the equipment was funded by the Wellcome Trust, the Biotechnology and Biological Sciences Research Council, and Dr. Tony Wild, an early York chemistry graduate and generous benefactor to his department, including, I understand, the pizzas which our younger guests tonight have eaten earlier this evening. Many thanks to Tony for that as well. And I'd also like to take the opportunity to thank others involved in making the building happen, specifically Johan Turkenberg, Duncan Bruce, Michael Plevin, Gideon Davis, Jamie Blaza, Keith Wilson, Lucy Hudson, and Simon Breeden. And with that, enough from me, and I'm going to hand over now, and I'm delighted to welcome Caroline Descent, Head of the Department of Chemistry, to introduce our speakers. Caroline, over to you. Thank you.
Hello. Thank you, Charlie, for that wonderful introduction to tonight's event. And I think it's very much a celebration event for us, a celebration of the new Dodson Building, but as well of, of Eleanor and Guy's contributions to YSBL and the wider YSBL community over so many years. So it's my absolute pleasure to introduce our two speakers for this evening, Dr. Richard Henderson and Dr. Jamie Blaza. Dr. Henderson is a molecular biologist and biophysicist who's an international expert in the field of electron microscopy. So Richard attended schools in the Scottish Borders regions and went on to study physics at the University of Edinburgh. He then chose to pursue a PhD at Corpus Christi College in Cambridge at the world-leading Medical Research Council Laboratory of Molecular Biology, where he worked then for most of his career, with the exception of some time that he spent in the USA at Yale University. Richard shared the Nobel Prize for Chemistry in 2017 with Jacques Dubochet and Joachim Frank for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. His exceptional cross-disciplinary work led to the development of new instruments that allow us to see the structure of biological systems at really unprecedented detail. And this is important because that insight into the structure is so crucial in leading to advances across the biological and biomedical sciences. Dr. Jamie Blaza is a senior lecturer in the York Structural Biology Laboratory, which is part of the Department of Chemistry. Jamie also studied at Cambridge for his PhD, which was focused on developing biophysical methods for measuring proton and electron transfer reactions that are important in biological systems. He subsequently embarked on a career development fellowship to learn about cryo-EM, and following further postdoctoral work in Cambridge, came to York to establish his own research group and bring expertise on cryo-EM into the York Structural Biology Laboratory. So thank you both for speaking tonight. It's our honour to have you both speaking tonight. And I'm now going to hand over to Dr. Henderson, who's going to speak first.
Thank you so much, Caroline. So it's a great pleasure to be here to celebrate the opening of the Eleanor and Guy Dodson Building. And obviously, I've known both Eleanor and Guy for more than 50 years, actually, since all of us started out as X-ray crystallographers in the early days of protein crystallography. So I thought I'd show 2 or 3 photos before I tell you anything about structural biology and science before we start. And so the first one is an early picture of the really very early day pioneers in founding protein crystallography. So in the middle is Dorothy Hodgkin. She was the group leader when Eleanor and Guy came from New Zealand to learn about and do protein crystallography here. And Dorothy had a Nobel Prize in 1964, probably a year or 2 after they arrived, for her work earlier on penicillin and vitamin B12. But she was one of the founders, took the first X-ray diffraction patterns of protein crystals back in 1934, when she was working in the lab of J.D. Bernal, who's the guy on the left, stroking his chin. So they were 2 of the earliest pioneers, actually. And then there's John Kendrew, looking on, and David Phillips. They were sort of second generation, if you like, in terms of getting into the field. And they're looking at the structure in 1964 of the enzyme lysozyme, which was the second protein structure determined. So I thought that was a good, before Eleanor and Guy. And then the next one is a lovely picture of Guy chatting to Dorothy Hodgkin in the 70s when they were working out the structure of insulin. And this was in the biography that Eleanor wrote about Guy. It's one of the photographs in that, really well worth looking. And then fast forwarding a bit, this was the group 34 years ago here in York that had been built up by Eleanor and Guy, starting from 0, or starting from themselves. And there was about probably 30 people there, and now you've got a whole department, and with a building of its own now. So that's a little bit of background and history. So then I thought, for the broader audience, I ought to try and relate what we're trying to do now with electron cryo-microscopy to how things started. So there are different methods. X-ray diffraction is the one I just mentioned, experimental way of determining protein structures. But before that, before there was the power of X-ray crystallography, people did do model building to find out how biological structures might exist. And then there's also a period, NMR is still going strong, but was for 20 years or so, also a primary method of determining protein structures. So these were the 3, and then following on, we have electron microscopy, electron cryo-microscopy, which can be used to determine structures that you can't crystallize or are too big for NMR, or definitely too big for model building. But I thought I'd show a couple of slides about each of these things before we start. So one of the most famous is the work that led to knowledge of the DNA, the double helical structure of DNA with its information about what the gene is and how you replicate and how you translate it to make proteins. And that this is Rosalind Franklin on the right, and her PhD student, Raymond Gosling, took this picture, Photo 51, which is quite famous. There's even a play that was on in London called Photo 51. And then with that information about the geometry of the structure of the double helix of DNA and other information, Watson and Crick built this model that you see on the bottom right, which was first illustrated by Francis Crick's wife, Odile, in that diagram. So that was probably one of the earliest and probably had the greatest impact in terms of early structured biology. And then another one was Linus Pauling, who won the Nobel Prize for chemistry in 1954, partly for his work working out, essentially guessing by model building, one of the elements that are present in the structure of proteins, which they did by analyzing X-ray structures of small amino acids and dipeptides, and Pauling knew that when you couple together two amino acids, they make a peptide bond that's planar, and then used that constraint, which other people hadn't realized, to build this model of alpha helix, beta sheets, and so on. And so that's a common feature in proteins. So that's nucleic acids and proteins, before there were powerful experimental methods. So then crystallography came into its own. This is a picture of sperm whale myoglobin crystals from John Kendrew's work, and Kendrew, remember, is the one standing between Dorothy and J.D. Purnell in that photograph, and Max Perutz, who started off as a PhD student with Bernal, but then Bragg took over in Cambridge, and they were the two in Cambridge pioneers of X-ray crystallography, and with diffraction patterns like the one shown in the top right. John Kendrew's group, it was a smaller protein than hemoglobin that Perutz worked on. They got this structure in 1957, 5.5 angstrom resolution, very sort of blobby. It was made out of plasticine, but these sausage shapes, they're about 10 angstroms in diameter, 10 angstroms separated from the next one, and those are the alpha helices that Pauling had predicted about 5 or 6 years earlier. So found in the first proteins and now found in hundreds of thousands of proteins. Okay, so then they went on in the late 50s, and by 1960, they had an atomic model of the first protein structure. This is 4 years before that picture of lysozyme. And there was no molecular graphics, high-resolution TV screens then, so they got an artist, Irving Geis, to paint this watercolor picture, which was the middle, the sort of center page spread in Scientific American in 1962. And that was sort of the beginnings of protein crystallography then through X-ray experimental work. And then fast forward about another 25 years and so on. And this is Jacques Dubochet that shared this 2017 Chemistry Nobel Prize. And he was hired by John Kendrew to be a young group leader in Heidelberg in 1978. And the job, or his target, which was agreed, of course, between him, was that they should try to develop electron microscopy methods, both cryo-microscopy, where you were cooling the sample. They built a liquid helium temperature microscope, and ways of studying the behavior of water so that you could look at it when it was cold in the electron. And this is Dubochet's apparatus. I'm not sure, yes. So here at the tip of the pair of forceps is a 3mm electron microscope grid, and Dubochet and Mark Adrian and Alistair McDowell's method was to apply a little droplet, 1 or 2 microliters, to that grid, blot it with filter paper, and then plunge it under gravity from there down into this little chamber with liquid ethane at roughly the temperature of liquid nitrogen, where it would freeze the water, the thin film of water, very rapidly, so you didn't get crystals like you get in snowflakes. And that amorphous film of water or buffer could have molecules, any kind of biological molecules, so ribosomes, viruses, and so on. And one of the first images they took was this one in 1984, was adenovirus, about 1,000 angstroms in diameter. It's an icosahedral virus, and you can see this thin film. The material in between the viruses is just a thin film of buffer, and then most of the viruses, 20 out of, and there's 2 or 3 that have fallen apart because this blotting procedure brings an air-water interface closer and closer to the molecules, and that's still one of the main problems. But that was 1984, and the computers weren't very powerful, the programs hadn't been written yet, and it took them 6 or 7 years to go from that 2-dimensional image to this 3D structure here, which is a sort of blobby structure, 30 to 40 angstroms resolution. You could see the subunits, but not much more, and then it took about another 20 years to go from images like that to images like the one on the right now. And you can see now that there's more detail in the image, and that was a higher voltage microscope with a much brighter, 1,000 times brighter electron source that gave you coherence, so it didn't go fuzzy when you tried to bring it into focus. And then those images like that produced maybe 15 years ago now the first high-ish resolution structures of biological material by single particle electron microscopy, which means, and then the next slide shows you an example from about that period of what you could do before we had the most recent hardware equipment improvements. And this is a sort of a good snapshot to say what is electron cryo-microscopy and why is it such a powerful method going forward and essentially out-trumping, if you like, X-ray and NMR methods. So what this one shows is 4 different types of specimen, all of which you can put into the same microscope, use the same equipment. And the one on the top left is a thin film of Dubochet method with E. coli intact 70S ribosomes, the site of protein synthesis. And that was the work of Joachim Frank, that was also sharing this 2017 Nobel Prize. And in the year 2000, which was when this is a figure out of a review that Tim Baker and I wrote for international tables for crystallography to try to explain why cryo-EM might be something to keep an eye on in terms of developing in the future, but it wasn't all that powerful then. So this is an 11 angstrom structure, not really good enough to resolve the alpha helices if there were any, but it was enough to resolve the large and the small subunit, tRNA, and some longer pieces of RNA. That was state of the art, say 23 years ago. The second panel is single particles, again in the thin film of amorphous ice, with symmetry. So this is icosahedral symmetry. This is hepatitis B virus cores. And this was work done by Bettina Botcher, who was a postdoc from Germany who came to Cambridge and worked with Tony Crowther. And she was keen to try and see alpha helices in order to get higher resolution without having to have crystals or without having to do NMR. And she got this structure at 7 angstrom resolution, and you can see these protrusions that are sticking out from the surface. And each of those protrusions is a bundle of 4 of Pauling's alpha helices sticking out from the surface, and they even built a model using their knowledge from the sequence and from labeling at that stage. So that was the first, if you like, an atomic model made by single particle electron cryo-microscopy of a biological structure. And then the other 2 have higher levels of organization. So the third panel is a one-dimensionally ordered structure, which is the thin filaments from muscle with the head group of myosin from the thick filaments added to it in the absence of ATP. So it's essentially like a muscle that's in rigor with no metabolism going on. And Ron Milligan worked out this structure at about 30 angstrom resolution. Of course, now all of these structures are at much higher resolution, but even then, by doing different SMAPs, doing a thin filament with tropomyosin, thin filament with tropomyosin and actin, they could find out and color differently the different components of it. And that helped them to understand, even 23 years ago or so, how the thin filaments and thick filaments in muscle pulled against one another. Now there are more high-resolution atomic models. And then the last one is an image of a 2-dimensional crystal of a membrane protein, the light harvesting complex from green plants, which is the main green pigment in grass and trees when you look around. And this was Werner Kuhlbrandt's work. He was the successor to Dubochet at the EMBL, which has a 9-year tenure, and then you're supposed to go off and make your way in the big world. And so that was Werner Kuhlbrandt's work, making crystals of the light harvesting complex, and that has 3 transmembrane helices and chlorophylls, 13 or 14 chlorophylls. And because it was a 2D crystal, they got a structure using electron microscopy at 3.4 angstrom resolution, whereas the single particle work was still really trying hard to do better. So then, over time, the microscopes improved, the vacuum, the brightness of the sources, and particularly the electron detectors improved. And so here is one of the first structures done using essentially current. It was 2014, but you know, the current equipment is not a lot different. It shows a picture of a field of view of ribosomes that couldn't be crystallized because Alexei Amunds couldn't purify mitochondrial ribosomes enough to get crystals. They always had 20 or 30% impurities, cytoplasmic ribosomes, but using software that Georges Cherez had developed probably about 10 years ago now, and working with Xiaochen Bai, who was an electron microscopy expert, they got first a blurry structure, and then using methods of classifying to separate out which ribosomes were the ones they wanted, mitochondrial ribosomes, which ones were cytoplasmic. They got images like that, and then with other types of refinement where you can disentangle different structural states, eventually they get a high-resolution model of the whole ribosome, both the large subunit and small subunit, from one set of images that takes them a day or so to collect. And then a little bit of the map. So this is, in A is a little bit of Watson-Crick RNA double helix with the 3 base pairs that you see here, and then a top view of one of the base pairs of the Watson-Crick type, same as in that model from the model building back in the 50s, with the 3 Watson-Crick base pairs between the bases, and then an alpha helix, exactly the same geometry as Pauling, and then a couple of turns with a magnesium ion. And so that was a really good example of a structure that still cannot be done by any other method. And then a second one is mitochondrial complex 1 that Jamie Blaza, who will be talking a little bit later, worked on in Cambridge, but this was 2014 again. So Judy Hurst, who's now the director of the MRC mitochondrial biology unit in a different MRC unit in the university in Cambridge, brought this sample over and said she'd heard cryo-EM might be able to do something with this kind of sample. And within a few weeks, Vinoth Kumar, working together with Jaipen Ju in Judy's group, came up with this map at lowish resolution. It's got 8 iron-sulfur complexes. These are the brown molecules. NADH puts an electron in at the top, goes down that chain of iron-sulfur and reduces a quinone in the membrane, and then other structural changes create a membrane potential through proton pumping, and then that's used to make ATP, and then your whole mitochondria and your whole body goes. But also in this picture, you can see that there's a region with 76 transmembrane alpha helical segments of the protein, and then at a lower contour level, the detergent that you use for extracting the membrane protein, and then with a bit more work, an atomic model with 45 different subunits, all built nicely into that map without any crystals and with a minimum of, well, a year or so of work. And then the core subunits, the blue ones, they had been solved earlier by another group leader in that same MRC unit, Leo Sassanov, who had crystallized a bacterial homologue that was thermophilic and then was good enough to make crystals, whereas they never got crystals of the mammalian mitochondrial one. Okay, so that's the 2 structures that I think give you a good flavor of why cryo-EM can do things that you can't do by any other method. And then if we look at now what's been done since 2014, the last 10 years now, these are the protein data bank deposits of atomic models of structures done by the 3 different methods, NMR, X-ray, and crystallography. And you can see that NMR was quite popular, say, 20 years ago for small structures that you couldn't crystallize. And it rose at about a quarter of the rate of X-ray crystallography and peaked in 2007. And now NMR is still very powerful, and you have it here, but it's used for, say, weak ligand binding or kinetics or mobility and so on. No longer so much for de novo, a little bit, but not for de novo structure determinants. X-ray crystallography grew and grew and grew, and then since the last 10 years or so, it's more or less plateaued. And then this little peak here, that was 2021, that was the coronavirus peak, when we were all told, you're not allowed to go to work in your lab unless you work on coronavirus. So suddenly you've got all these people trying to work on difficult projects, suddenly all working, and then there were about 5,000 structures, many of them very similar. So there was a peak there, and then everybody, of course, went back to their normal work. But if you compare now, and then the electron cryo-microscopy, this is still in exponential growth. So whereas NMR has peaked and is declining, X-ray has got to a plateau, and now people are being more and more bold, and the microscopes get easier and easier to use. So this is the plot that we like to look at, showing you that 20 years ago there was 1,000 times more structures per year done by X-ray crystallography than by cryo-EM, and then the hardware improved, and then there was about 200 times as many per year. But since 2013 or so, there has been, of course, in that year, there were only 3 labs in the world that had equipment good enough to do those structures. But gradually, people have bought more, and there's now hundreds and hundreds of these. And here in York, there is one of these new high-resolution microscopes, the Glacios. And I think, obviously, York may be thinking of having more. But anyway, it's grown and grown and grown. And so now, worldwide, it's about 2 to 1. You can see 1.98. So this year, there'll be 10,000 structures, roughly, deposited by X-ray crystallography and 5,000 by electron microscopy. But we think it's going to keep going. Certain subtypes of structure, like membrane proteins, or certain labs that have been ahead of it, they're already doing 10 times as many structures using cryo-EM as they are by X-ray. So we think it's pretty safe for at least 10 years that it'll be growing. But eventually, you need to tackle what will be the rate limiting step that stops it from being exponential growth. Just like in the computer industry, they look, they've done 50 years of doubling every 2 years, and the EM's only done 10 years. And they've gone up 20 million fold in their number of transistors per square millimeter or something, whereas the EM still got a long way to go to catch up. So then I thought so that's where we've got to, that's why it's so good. And I thought I'd just show you 2 more things. One is, what was it that we did in Cambridge? Well, early on, Nigel Unwin and Nigel was a New Zealander also, and he came to Cambridge to do a PhD in metallurgy, but he was recruited to bring his knowledge and skill into biology. And he was giving a talk in 1973. I was supposed to be working on membrane proteins with X-ray. He was doing this on viruses and so on. So, but anyway, we worked together for about 2 years and managed to get this, in 1975, this model of the first membrane protein that there was a model for, with 7 transmembrane helices of the Pauling type, and then with about another 15 years of work, we managed to get it to higher resolution, and this is a phenylalanine, a tyrosine, different side chains. So that's what we were doing, and that led us led me from X-ray into electron crystallography, and eventually into imaging and single particles, no crystals anymore, because it turns out the images you can get are just as good as the diffraction patterns. So probably about 7 or 8 years ago, Vinoth Kumar and I, Vinoth was the one who worked on the mitochondrial complex 1 initially in Cambridge with Judy Hurst's group. We wrote a review, which was written in 2015 now, about 8 years ago, saying that this was all very good and fine, but we needed more microscopes so that it wouldn't be so expensive. There are £5 or £6 million for one of these high-end microscopes. So we said, okay, we need some cheaper ones. And the original plan was just to get them to be cheaper. We tried this out on the companies, and they said, well, no, we don't really like cheap microscopes. We like expensive microscopes. So we said, okay, we have to go ahead, and our plan was 100 kV with a field emission gun. And just at that moment, someone came through the lab, one of the Thermo Fisher, microscope manufacturer. He was doing some private work. He said, I know somebody in York who makes field emission guns might be just right for your microscope. This is Mohammed El-Gamati, who's here, it's row 4. So anyway, we got together with Mohammed and with an X-ray detector company that made X-ray detectors that could be useful. And this was the paper. This is Matthew Peat, who's a postdoc, and it shows a plot of the inelastic and elastic cross-sections versus the electron energy. And we knew about this, but we weren't really sure it was true. But Matthew measured these cross-sections, and indeed, if you look at these, this is on a log scale vertically and also horizontally, and the experimental measurements fit exactly on the theory. And the hidden thing behind it is that when you bring the voltage down from high voltage, 300 kilovolt electrons, which we said we must have this because of other reasons, bring it down to 100 kV, you actually get not only a less expensive microscope, but a better microscope. And so then the companies said, oh, well, do we really have to make, anyway, they're sort of moving in the right direction, but not fast enough. So we got a grant from the Wellcome Trust, and this was the plan, a gel microscope column that costs about £200,000, a York probe sources field emission gun, similar, and a dexterous detector, similar, with side entry stages. And we built this microscope, and then Mohammed just told me in the pre-talks drinks that this paper, this is figure 1 in the paper, is now just accepted in PNAS, so you can look it up in a few weeks. But anyway, it has the gel column, the York probe sources field emission gun, and the dexterous detector. Here's how it looks in reality. It's got an image of graphitized carbon on the screen. These are the side entry cold stages that we use to put the sample in. Here is an image of gold. And the detector is not perfect. We need new, bigger and better detectors. But this is an image of gold on carbon, and these are the diffraction patterns from the image at 2.3 and 2.0 angstroms, and you can do a little bit better than that. And then we thought, well, we better, to encourage the companies to be bolder in their vision, this is what's in the paper. There's 11 structures between 2.5 and 3.5 angstrom resolution, including one. This is acetoxy hydroxy isomer reductase here. This one, the last time I was in York in February, we came to be given a specimen of AHIR, which was made by Olivia Gittins, who's a PhD student in Newcastle, here in February in York. And we took the grids back to Cambridge, put one of them in the microscope, collected data for a day, and you get this structure. And then the other ones were specimens that we had or other people provided. And you can see from this table now, they're all between 2.5 and 3.5 angstroms. So really quite good, still not quite as good as the 5 to 10 million pound microscopes, but we're kind of working on that. Here's some density, alpha helix. Here's a ligand, etomidate bound to a glutamate ion channel. And then I thought I'd finish just by showing some of the people that I've personally worked with. The key one was Nigel Unwin, who is the one who converted me from being X-ray crystallographer to electron crystallography, and then eventually to, so the bacteria Dodson collaborators are in the second row, the detector people are in the third row, and the single particle people, which is this sort of evolution of the field, in the third row. And then currently, I'm mainly working with Chris Russo, who's a new young group leader with Matthew Peat, was a postdoc with him. And so we've got a program to go forward and make the cryo-EM sufficiently user-friendly and inexpensive that every student can have their own microscope. That's the plan, anyway. Okay, so thank you very much, and over to Jamie. Yeah.
Thank you, Richard. There we go. Hello, Vice-Chancellor's already introduced me. I'm Jamie Blaza, and I'm the academic lead on the cryo-EM project at York. And I'm going to be talking a bit more specifically and a bit more about the present and the future, with Richard covering the past, about the Eleanor and Guy Dodson Building, and what we're trying to do with it and where we want to take it and what it allows. But first, I have to introduce Eleanor and Guy. And so they're an incredible team. Here they are on their wedding day. This is the one photo they took on their wedding day, which also tells you how organized they are, in Oxford, and they're working for Dorothy Hodgkin, where they met. And they moved up to York in the late 70s to found YSBL. Here they are in the early noughties in the biology department atrium. As the VCs already touched upon, sadly, Guy passed away in 2012. So I never met him, I didn't know him, but certainly everyone who speaks about him always has such positive memories. And what was really important was that Guy and Eleanor were an incredible team. Particularly, Guy was fantastically good at recognizing talent in others and getting the most out of them, although Eleanor's also pretty good at that. And Guy was really a builder and a founder, so he founded YSBL. He then went on to found crystallography at Mill Hill, which became the Crick Institute. Eleanor is fantastic at the very, very fearsome maths that underlies protein crystallography. And so the pair of them were fantastic scientific founders for us. But the other heritage they've left us is that they also established a very positive culture in YSBL. And in the same way that a negative culture can become self-reinforcing, a very positive culture also becomes very reinforcing because people are respectful and generous with each other and share ideas and support each other. And we still benefit very greatly from that culture today. And just as a little anecdote, here I am cycling into work this morning, and I bumped into Eleanor, walking her dog. Her first question was asking me how my son was, which shows you she's always got people's children on the front of her mind. And the next question was whether I knew enough about the programming language Python to help her, because Python was broken, and that means she wasn't going to get any work done for the rest of the day. So before we talk a little bit more about length scales in biology, I wanted to introduce length scales and sort of particularly for the younger members of the audience, remind them that you have an organism. I've chosen a picture of my son to illustrate an organism. And then below an organism, you have organs. And then below organs, you have tissues, and then cells, and then organelles and chromosomes, and then enzymes and other biomolecules, and finally atoms in biological molecules. And if a biologist wants to study things on this scale, that's fairly easy because you can just use your eyes. If you want to study things on this scale, then you can use a light microscope, but a light microscope is limited by the wavelength of light at a few hundred nanometers. If you want to move into this scale, you need structural methods. And so this is the roots of YSBL, the York Structural Biology Laboratory, is crystallographic methods, and particularly macromolecular crystallography. And here, you start by growing a crystal. You don't get enough information if you hit a single protein molecule with an X-ray. So you need to grow a crystal to amplify the signal. You then collect a diffraction pattern, and these can be quite stunning, like this one here. And you collect a diffraction pattern, and that diffraction pattern arises from the planes within the crystal. And by solving the phase problem, you can then reconstruct the three-dimensional model of the molecule that gives rise to the crystal. And as much as I've just said here, oh, well, you'll solve the phase problem, actually solving the phase problem is enormously difficult. And particularly in the early days of crystallography, people could collect many diffraction patterns, but they couldn't solve the phase problem, and therefore they couldn't get the information they needed to build a structure like this and to zoom in on the bits that mattered so they could understand how biological action came from a three-dimensional structure. And so this is really the roots of YSBL, people sitting around computers, scratching their heads, trying to get the most out of these very information-rich patterns and to convert them into something really, really useful. And YSBL was a center for crystallographic methods. And this was really nicely awarded by the Queen's Anniversary Prize. There's Charlie picking up the prize with Tony Wilkinson, the head of YSBL. The nice thing about the Queen's Anniversary Prize is it's one of the very few prizes that's for groups rather than for individuals. So it's very fitting that that was given to YSBL. So there are three main structural methods, and Richard's already introduced them, so I'm not going to go into a huge amount of detail here. The first one's macromolecular X-ray crystallography, which I've just taken you through, and that's how you get to structural information using macromolecular crystallography. The other one is cryogenic electron microscopy, where you use an electron microscope and a vitrified sample, and by averaging over many thousands of particles, you get to a three-dimensional map, and I picked complex one, which Richard's also introduced. because I worked on that after Xiaopeng and VNOth in Cambridge, and actually it was a project I was very happy to leave behind because it was a nightmare. Finally, NMR is one that we don't speak, that hasn't been introduced already in much detail. Here you place the sample in a superconducting magnet, use radio waves to interrogate the atomic nuclei, and then you can generate from those signals something to generate a structure, but also, as Richard's already introduced, it's now more used for looking at ligand binding and other things, looking at what proteins do rather than their intrinsic structure. But there's a few observations about all of these three techniques. Firstly, they all rely on very large pieces of capital equipment that will set you back at least £1 million, if not many millions of pounds. And that means they all have to be put in perfect location, and they all have to be used very efficiently, and these pieces of capital equipment become really the centerpieces of communities who try and make the most of them. The other sort of personal observation you have is, of course, that these patterns that you get in X-ray crystals, they're pretty inscrutable. They look very complex. You don't really know what that means without a very detailed analysis. Same with NMR. With EM, you think, oh, fantastic. You're just, okay, we're using electrons rather than light, but at least we're now getting normal images. But if you actually start digging into the contrast formation mechanisms in a microscope, they're rather deceptive, and you start digging into contrast transfer functions and all sorts of very advanced maths to do high-resolution reconstructions. And the other observation I have here is really all of these techniques are the meeting point of the physical sciences, the biological sciences, and maths. And now in modern universities, there's a big push to do multi or interdisciplinary research. With structural biology, the simple reality was we had to be interdisciplinary from the very, very earliest days of the techniques in the 50s and the 60s. So now I've kind of introduced the type of work we're trying to do. I can say here is the Eleanor and Guy Dodson Building, in a sunny day, unlike today. And the VCs already thanked the donors who made it possible, and particularly Tony Wild, who was an early chemistry graduate who's now done a huge amount to support the university and the Department of Chemistry. In terms of seeing a project like this through, here's the York team, of course, the Estates Department were a huge help. They had to be. The chemistry head of department was Duncan Bruce. I don't think he knew what he was getting himself in for when he began his tenure and took this on. The university lead on the technical side of things was Johan Turkenberg, and we'll come back to Johan's huge contributions in just a moment. The PI on the cryo-EM grant who wrote it and planned it was Gideon Davies. The lead on cryo-EM was me once I was recruited. I definitely didn't know what I was getting myself in for. And the lead on the NMR side of the building was Michael Plevin. So this is how the building looks, and you can see it isn't a huge building. The VC's already touched upon how expensive it is, and he's absolutely right, it is enormously expensive. And in many ways, it is just good to get all of your expensive equipment in one space and just have the space completely designed around doing these activities in the best possible environment that they can be done in. So we have 2 prep labs held at 2 different humidities. We have an NMR lab, which has to have very, very low stray fields. And here we have the cryo-EM lab, which has very careful airflow, very low humidity, and the microscope is on a very stable, massive cube of concrete that weighs many, many tons. Here, Sam and Johan sit. Sam's a senior technician who's worked in YSBL for at least 15 years and is a huge part of our success. And on the left, you have Johan, who really masterminded the technical sides of the building. And in particular, when a project like this was inevitably over budget, it was Johan who went into those meetings with a complete command of all of the different technical demands that we had to make sure that when we found the savings that we could make, we didn't compromise on performance. And now we've moved into the building and got some data out of it, we can see indeed that that was successfully done. So here's a few photos of it being built. We started with a wet and drizzly car park. This was a gas mains. At one point, we had a panic because we realized that apparently there was a gas mains where we wanted to put the foundations. We actually, when we finally made a plan with Northern Gas to move the gas mains, when we dug down, we realized that indeed at some point in the past, a gas main had been there, but it had been removed by someone else, and we don't quite know who, but there was no gas main when we got there. And then the foundations went here. And you can see where the cryo-EM sits on its plinth there and there, or there and there, and then the walls went up and we were in. And then in late 2020, here's the delivery of the microscope, which is the first bit of kit we moved into the building. Of course, we were all wearing masks because it was COVID time. So what have we done with it? And we haven't had it for a huge amount of time, but I just wanted to touch upon, I mean, just a whistle-top tour of the diversity of the projects that we've had going here. One project was looking at a bifurcating hydrogenase, an ion-ion hydrogenase, which takes an electron pair, splits it, sends them down 2 different routes, which is a really remarkable thing to do. Fred and Dorothy have been looking at DNA packaging into a viral motor. We've looked at the molecular basis of human lysosomal storage disorders. We've done, and this is Hugh Jenkins, who's led on actually doing stuff that isn't even biological by doing microelectron diffraction. I mean, for that, he has to stay up till the very early hours of the morning just collecting data. We've looked at the most abundant phage in the human gut. We've looked at how ribosomes hibernate. We've done a bit on sulfur sugars, and Pavel's even isolated a virus that he's called Jorvik, after the Viking name for York. But for the case study, I thought, particularly if we have Richard from the Medical Research Council, I thought I'd pick something which is a collaboration between YSBL and the Centre for Novel Agricultural Products, partly because this was in the plant science and sustainability space, partly because it's just a really nice example of how, when you have 2 excellent research centres on campus, they can work together to have even more effect. Now, I just mentioned this paper actually included other contributions which weren't cryo-EM based from those people, particularly Michael and Clement, who used the NMR machine, and that went in. So the science here is around the fixation of carbon dioxide by algae, and this is an important and really difficult reaction, because of course carbon dioxide is a greenhouse gas. If we could fix it better, then we could take carbon dioxide out the atmosphere and fight climate change. And the way that most organisms do it is they take carbon dioxide, they combine it with this sugar to make 2-glycerate 3-phosphate, which is then used by metabolism to build the cells. The enzyme that does this is Rubisco. This fixes about 30% of global CO2. Specifically, algae fix 30% of global CO2. It's not just plants. And chemically, this is a thermodynamically and kinetically demanding reaction. So what does that mean? Thermodynamically demanding reactions are ones where it's very hard to find conditions where the reaction will even proceed in the first place. Kinetically demanding reactions are reactions where even if you've created conditions where the reaction will proceed, it won't go very fast. And we're going to come back to how the organism gets around these difficulties in just a moment. But first, I want to introduce a model organism, algae, Chlamydomonas reinhardti, that James Barrett was working on as a PhD student in Luke Mackinder's lab, who I introduced a few slides away. And I actually really like this movie because you can see here my 3 main modes of working are either sometimes I have energy and I get places, sometimes I have no energy and I just sit there, or sometimes I'm like one of these guys and I just sort of have tons of energy, but I just spend it spinning around, not getting anywhere. But if you put an algae like this into an electron microscope, you can see this bit of the algae called labeled P, which is the pyrenoid. So the pyrenoid gets around the problems with fixing carbon dioxide by firstly having an enclosed space that the organism just pumps carbon dioxide into. And that gets around the thermodynamic difficulty of the reaction, because by putting in lots of carbon dioxide, you push the reaction over to the other side. And then the other thing with the pyrenoid is it's just incredibly densely packed with Rubisco. And this was the question James was asking with the bioinformatic pipeline. How is that Rubisco packed so densely? So this was our experimental workflow. James started by isolating these Rubisco enzymes. He actually used a slightly different species. He used chlorella, and he had identified some linkers that he thought were allowing the Rubisco to be pulled together into this tight condensate. And so we isolated Rubisco, we combined it with a purified linker, and we prepared vitreous ice grids. And then we imaged them using the glaciist, and we ended up averaging together thousands of these particles, and you can now see these fuzzy shapes now become much more detailed 2-dimensional images. And for this, we used the York Viking cluster. We use Viking 1, and we'll use Viking 2 when it's operational. But then we could go to 3D, And when we went to 3D, we immediately recognized the diamond in the middle is the Rubisco molecule, but around the edge of the diamond are these funny little bits of density, which are actually the linkers, which are sticking to the side. So I'm not going to go into the details of how we did this, but because the linkers were not bound to all of the sites, we had to use symmetry expansion to get around the Rubisco symmetry, and we could end up with a clear model for how the linker interacts with Rubisco. And that gives us a molecular basis for understanding this interaction, which will allow in the future engineering of the interaction to either make it stronger or weaker, depending on what we want to do with it. The other thing it's clear from the fact that it wasn't all bound is the linker has 8 weak binding sites rather than a single strong one. And this is really relevant because X-ray crystallography would have really struggled with this because crystals need something that's completely similar, so they'll all crystallize together to make a crystal. But if you have got linkers binding to some of the sites but not some of the ones, you don't have identical subunits that all come together to make a crystal. Finally, and maybe most excitingly, the linker is very, very simple. And this is going to make it easy to genetically transplant this into plants. And Luke McKinder's got a grant from the Carbon Technology Research Foundation to start exploring doing exactly this. So I just want to talk for 5 minutes on the future and where we want to go, and that's electron tomography. So really, more than the last 100 years of biochemistry has been spent grinding up cells and looking at the bits, or taking the genes for our targets and overexpressing them in E. coli. And it's been very productive, and structural biology is certainly no exception to this paradigm. But increasingly, we understand the bits better and better and better, but we don't really understand how they come together to make a living cell because we don't have a structure of the cell. Single particle analysis, which is how we studied the Rubisco, is all based around the idea that the particles we image are identical, so we can average them together. But a cell isn't like that. A cell isn't regular. It might have similar structures, but they won't have exactly the same conformation as each other. So we need tomography. How does tomography work? So this is a nice example, actually just from Wikipedia, of rotating a 1D detector around a known 2D shape, which is this pi shape. And you can see this movie goes round, and these rays, which could be photons, they could be electrons, they could be X-rays, as they go through, they make an impulse on the detector. And I think as it went around, you can kind of see how each one of those gives rise to an impulse on the detector like that, particularly when it's lined up on the flat bits like that. But what if we don't know our structure? What if we have an unknown structure? And so here, we can do back projection, and by sampling around something, we can reconstruct its shape. So that's a 1D detector around an unknown 2D shape. Here, we're rotating a 2D detector around an unknown 3D shape. So this is actually a pyrenoid that we've reconstituted in vitro using isolated components. Sabina Musial's doing this work. She's a joint PhD student with Luke and me, with a lot of help from Pavel Bardi, who's a Sir Henry Wellcome Fellow we're very lucky to have in YSBL, who's just trying to establish and build tomography. So we have to do in vitro because we don't have a high enough voltage microscope and we don't have other bits that we need. But can you apply this to a real cell? And actually, here's one of the real first proof of principle studies that were done a few years ago on Callidium ammonis, looking at the pyrenoid. And you can see a huge amount of molecular detail. This scale bar is just 200 nanometers, and we can build a three-dimensional map of the pyrenoid on the most basic molecular level. We can see the green are the membranes that are used to pump carbon dioxide into the pyrenoid. We can see in purple all the different Rubisco molecules, how densely packed. We can even see these extra little tubules that run along the middle. The reason that these workers could do this is because they had an energy filter. So particularly with thick samples, a lot of your electrons will become scattered, and they'll become scattered inelastically. So we need to filter them out. And so this is approximately how this works. This is a sort of simple diagram of electron microscope. After the sample, we have these inelastically scattered electrons. We want to filter them out using an energy filter. Like everything else in EM, it's hideously expensive, so the energy filter costs about the same as one of Richard's 100 kV systems that he's developing with Mohammed. But just on my final slide, I want to talk about building up cryogenic electron tomography at York and how do we get this going. And the reality in experimental science, you're going to need three things. The first is you need equipment, you need space to put it in, and you need people who know how to run it and what to do with the data. So the equipment is in progress because I'm sitting in my office writing grants. The space to put it in, of course, we have a beautiful building. Here's Eleanor cutting a ribbon earlier today. We have the Dodson Building to house all of this equipment. But the other thing is we have people who know how to run it and what to do with the data. And this is really taking YSBL back to its roots, because tomography's burst onto the scene and we can collect a lot of very complex data of cells at the really basic molecular level. But a little bit like in the early days of X-ray crystallography, where you could get a diffraction pattern and if you knew how to get data out of it, have something to interpret. But if you don't have anything. And here we have, we're going back to our kind of roots of people sitting around computers trying to understand their data. And this is, the only thing that's really changed is that the computers have got a lot more powerful. So that's me done. This is all of YSBL outside of the Eleanor and Guy Dodson building. We're very proud of it. We're very grateful to you, Eleanor, and to Guy. You set the culture. You set us going. We wouldn't be here without you. And thank you all for your attention. Cheers. Yeah. That sounds good.
So I think we have all thanked Richard and Jamie for their wonderful talks. We have probably about 5 minutes for some questions. So I'm going to ask Richard to come and join Jamie and I at the front. I'm going to sit in the middle. And I would encourage you, if any of you have any questions, please do raise your hand and somebody will come with a microphone. So is there anybody who would like to start us off with either a question or a comment? It's hard to spot in here, isn't it? There, we have a question.
Thank you very much for that. It was fascinating and amazing to know that this groundbreaking work is going on so close to where I live, where many of us live. I feel almost uncomfortable asking this question because it's cliched, I think, but artificial intelligence, what's the scope for using this to accelerate the kind of work that you're doing? And is that going to make a big difference to how you start to develop things over the coming year or years?
Do you want to go?
Yeah. So, and particularly, I think many people saw the publication of AlphaFold, which allows us to predict protein structures, and that's really, really exciting. And I think particularly, as I mentioned, our kind of background is computers. So John Aguirre in YSBL, as soon as AlphaFold came out, was one of the first people to actually get it running. It's not that easy to get it running locally, and we can't just rely on the kind of database that they have. So yeah, enormous. And even before artificial intelligence, which is quite an undefined term, one of the things that YSBL developed a lot of with Catherine Kautan was trying to build models better, because the maps you have, if you're in a perfect world, will be atomic resolution and you just know exactly where everything is. But in practice, you never have data that good. So you have to interpret it in some ways, bringing in what more you know about the basic geometry of stuff. And artificial intelligence allows you to do that much, much faster. So it allows you to move much faster. But particularly the kind of AlphaFold models that come out only work for the protein parts of the molecule, or the DNA bits of the molecule. But quite often you'll be interested in small molecules binding or unbinding, or the location of the waters, and it can't predict that because that's not in its language. And so you kind of use the artificial intelligence to solve your structures quicker, particularly if you're doing molecular replacement in crystallography. And so I think that's the key. It allows you to move faster rather than do anything at the moment that I would say is fundamentally different.
Hi, thank you. That was a really good talk and very eye-opening. Where do you think the final place for this technology is going to be? Where do you think it's going to finally end up? Where's it going to stop? What is the end game, for lack of a better word?
It's definitely you.
Yeah.
Yeah. So, I think a lot of people are interested in, what are the current barriers to further progress. So most scientists are either looking for new ways to use existing technology, applying them to new problems. But other people are saying, well, what we'd really like to know, for example, is how is it that you remember a telephone number? You know, let's say only 6 digits. You remember it forever. You know, you probably learned it, you know, your first telephone number, you remember it. And it's really unknown completely where that's stored. Somewhere in the brain. I think it's, you know, it's definitely not in the heart, it's in the brain. So, you know, I think we are looking forward. So my view, particularly on the cryo-EM, which is sort of partly one of the techniques in the new building and partly what we're focused on the next steps for. But eventually it'll become just part of the woodwork. It'll become a commodity. Like, you know, you want to make a cup of tea, you have an electric kettle, and no one worries about whether electric kettles will go out of fashion. There are, you know. So I think what will happen is that the cryo-EM hardware and the computing will just become part of the woodwork, really. You'll just be using it like you measure pH or you have your DNA sequenced and so on. It'll become, and then people will move to asking the next question, how do you find out how all the proteins interact? How do they do structural changes? And then eventually, once we know pretty good examples of all the structures in biology and all of how they connect themselves together, that's what exists. And these are discoveries. And I think one of the things I've noticed recently is a lot of scientific groups searching around for new things to discover aren't so excited about that. And so they then start to invent things. And even though there's a limit to what you can discover, there is no limit to the number of things you can invent. So, you know, you have mobile phones didn't exist, or smartphones didn't exist 20 years ago. So I think they'll be even smarter. And you can talk to them at the moment. And then artificial intelligence, ChatGPT, you know, there are sort of, let's say, they're aids to humans to make more progress, but also, you know, humanity is evolving. So I think you could, you know, in other words, there's no end point, it just goes on, and you can't, you know, you should only worry about things that are likely to be developed, let's say, in the next 100 years, and then leave it to the next generation to go after that.
Thank you. It's very, very interesting. There was a hand that went up, I know, yeah, just very, more or less the same time, so yeah.
Yeah, I have a question for Professor Henderson. You moved from physics to biology. How was that transition? How did that happen? And would you have any advice for other people doing the same and looking to move into interdisciplinary research?
Oh, yes, I can definitely encourage you to do physics. But, you know, when I was a physics undergraduate looking for something to do, I asked a similar question, actually, what would it... And I looked at all of the things like fusion research, single particle research, solid state. I decided biophysics was the thing, went to Cambridge, and I was interviewed by John Kendrew, the person on the first slide that I showed. And he didn't really have any questions. He didn't seem to worry. They were just hiring people. But he said, did I have any questions? And I had the same question. I said, how is it, you know, if you come from physics and maths, won't I have to learn some chemistry and biology? Mm-hmm. And he said, oh no, he said, I wouldn't bother with that. You can pick that up as you go along. So basically, I think that's true, you know, and also you can learn physics out of textbooks and so on. So I think if you just take a naive, intelligent person, they suck knowledge in and then they can ask other people to tell them things. And so I think, yeah, no, it shouldn't be a problem actually. You can switch fields later, several times in your career as well, so.
Yeah, and we'll have one last question. I know there was a hand that... Yeah.
It was a related question. I was going to ask what your advice is to the many school and college students who are here this evening.
You can go first, Joe. I'll do something after Jamie says. And maybe Caroline has.
Yeah. Possibly, yes. Go on, Jamie.
I don't really know what my advice would be. Obviously, it's all worked out quite well for me studying science at university, but then there's probably survivorship bias there, that maybe people who doesn't work out well for studying science at university, but they wouldn't be here, so you wouldn't hear their advice. But I certainly think that the world is facing a very, very challenging period in its history, and there's so many problems. Antibiotics are disappearing, you know, we've got climate change, and really a lot of the solutions, not all, like it's not all going to be science, But a lot of the solutions will come from science and from engineering, and I just think we need our best and brightest studying those kind of things. And I'd just encourage people to get into that.
Yeah, then I had 2 books that I thought were interesting when people are thinking about, careers and how to. So there was a guy called Peter Medawar, who was the director of an MRC unit at NIM, National Institute for Medical Research at Mill Hill. And he also wrote some sort of semi-philosophical books. And one of them was Science was the Art of the Soluble, where, politics is the art of the possible. So you can try to do anything, but if it's not possible, you have to. So soluble meant if you're doing scientific research, you shouldn't do something that's too simple, because it's easy to do and you can predict it. But also, if you look too far in the future, it's not possible. The techniques aren't there. So he said, you've got to time it right and pick topics of interest for research that can be solved at least now or in the near future. So that was, Science, the Art of the Soluble. And then his other one, he did have a book specifically called Advice to a Young Scientist, which is full of sort of gems of wisdom like that. So anyway, that was.
Yeah. No, I think that's a wonderful point to end on. I think science can give you an amazing life of, thinking about things and really following what you're interested in. And it's such a creative, I think, career to follow as well, which is something we don't say enough. But yeah, I think if you're interested, it's a wonderful place to go with your life. Yeah. So I think that was a great question, really, to end on, and probably I should, I'm going to hand back now to Charlie, who's going to do the vote of thanks. So I'm not going to say thank you again, but hand back to Charlie. So yeah, I think you can. I don't know if you're supposed to sit here or not.
Well, I have to say, I have been wowed by that. I'm a political scientist by background, so all the technical chemistry has kind of flown past me. But what I've seen and heard in this session is a wonderful and extraordinary story of science. Richard gave us a kind of genealogy of his subject and showing us the progress of scientific discovery in structural biology through different generations of scientists, each building on the insights of the last. We've had a version of that here in York, going back to Dorothy Hodgkin, connecting to Guy and Eleanor, and all of those others, in this room, Tony, Robin, and those who've carried on that tradition, and Jamie now taking that forward. And in that really, really short bit of the story that Jamie gave us from putting that building together and starting to do the work, those initial findings, the enormous potential of that work on algae, because if we can find ways of capturing more CO2 from the atmosphere in our plants using that technique, it will be utterly transformational in meeting that challenge of climate change. But it doesn't stop there, because there's the future tomography that you're now beginning to envisage. So that story will continue, as I think the last question kind of signalled. And who knows, it's been absolutely wonderful to have school and college students from across Yorkshire here tonight. Some of you, I hope, will be working in the Guy and Eleanor Dodson Building, or some building like that in some university somewhere in the world, pushing on that story of science. I think it's been an extraordinary set of conversations tonight. So thank you to Richard and Jamie for those brilliant presentations. Thanks to the questions from the audience. Thanks to Caroline for chairing the session. We are going to have a few final words this evening from Eleanor Dodson, who is going to add a few thoughts to this. But before I hand the microphone over to Eleanor, I have a presentation to make. Because you've been rather important to this story, Eleanor, and we have some extraordinary colleagues in our workshops who can make all sorts of things. So basically, Eleanor, in honour of your part in this story, we have insulin on a plinth. So this is the structure of insulin, which Eleanor and Guy helped us to understand much better and to get the uses out of it that can be made. So Eleanor, this is for you.
Which has some of the features, but is kind of a bit fragile, to say the least. I'm sorry to keep you all for your dinners and so on. I did think I needed to say just one or two things. First of all, thank you very much for this honour, but it's, I think the structural biology lab was a wonderful place to be, and I'm glad that it's acknowledged in this way. But it's more, to acknowledge that structural biology is much more than to put up a name like Dodson of the Building, because it was so much more than us. And also, I think it should have been Guy and Eleanor, because it was so much Guy's inspiration that, broad interest in the chemistry and biochemistry, but also in the technology that allow it to happen, which drove YSBL for so long. And I was part of that technology. I don't know if he married me because of that, but maybe, maybe I had other attributes, I don't know. Anyway, I wrote this out because I knew I'd get it wrong otherwise. I just said we were very lucky indeed, and this doesn't just apply to me, but to all of us who were in the world of what we called structural biology in the 80s and 90s. There was this explosion of the problems were interesting. The biologists were coming up with all sorts of questions which you couldn't answer directly. X-ray crystallography was, Dorothy Hodgkin said in the 1930s, it's a wonderful thing to be able to see the atoms. And X-ray crystallography had built a balsa wood model of insulin and so on, but you wouldn't exactly say you'd seen the atoms. And we knew that protein crystallography could help to answer some of those questions. And we also knew that if we got the answers, we'd be able to use them for useful things. The insulin structure did help design better insulins, better therapies for diabetes and so on. But in the 1980s, the technological developments were taking off, and we were so lucky to be there when this was happening. The data collection for getting those images that people put up, I remember, it took months to get a set of photographs, but suddenly better detectors, better generators were there, and you could hope to get a set, maybe even a week. And at the same time, the computing was coming through so wonderfully. And you can't imagine the excitement of the first graphic machines. We had that photograph that somebody put up of Guy and Dorothy with their paper maps sort of studying where every peak came. But suddenly, it was all there, and then it wasn't just in black and white, it was in colour. It was brilliant, it was just wonderful. And then the next thing, which was what PCR. I don't know if Jim Brannigan's here, but he said once if I said reciprocal lattice in a talk, he was going to say PCR. I'm not even sure what it stands for. But it meant that with PCR you could begin to, once you knew the sequence of a protein, you could begin to create it in the lab. And then once you created it yourself, you knew something about it, you begin to introduce sensible modifications which might make it more effective for whatever purpose you wanted to use it for. So we were very lucky to be here at that time, and the results poured out, but we mustn't forget that what we weren't interested, and this is still true, we're not absolutely interested in the snapshot of a stable molecule. We're interested in how those molecules work together to make a living cell, you know, and we're still a long way from answering that, but we're getting nearer and nearer, and some of those EM pictures of elaborate patterns of different subunits interacting means, you know, we're nearer than we were to understanding how we tick. I just want to thank some of the people who helped it flourish here. First, I want to thank the chemistry department because they didn't give Guy an appointment as a lecturer, which was, at the time, we were told there were people who thought that he'd never be much good at teaching the first year undergraduates. But anyway, somebody allowed him to come here. And then for providing the support as the lab grew, because they really had rather let it cuckoo into their nest, you know, and things got out of hand, as you might say, in the numbers field over the years. Then we have to thank the university administration, which in those early days when there was only 3,500 students, I think, were very much, I'm sure we can find a way to make it happen. You know, there was a lot of flexibility, there was a lot of generosity, and they were fantastic support. And then the other thing I think we must thank is that enlightened government idea of having new blood lectureships, that they would appoint a person to a lectureship. For 5 years, they were to concentrate on getting their research going, doing a bit of teaching, but not to give a full teaching load. And York benefited so much from that. Rod Hubbard was a new blood lecturer. Robin Peruts was a new blood lecturer. Keith Wilson and John Helliwell, who became leaders in the synchrotron field, were new blood lecturers. So we should be very grateful to that initiative, and I hope that there's, at least now in the university administration, people are still helping young people get their research going. And then, of course, we were extremely lucky because insulin was obviously an important drug. There was still a big market for insulin in the world. And the Danish pharmaceutical company Novo had given Dorothy their first samples of insulin, and they continued to support the research in York as well. And that sort of, Guy called it slushies, so it wasn't quite slushies because you were meant to deliver something for it. But it was money that was available, and it helped get the equipment installed. We were very, very lucky in that. But the other thing I'd like to emphasise, and it's true for as now as then, the success of the lab depended on the fact that we all worked together, and that, you know, we knew we couldn't analyse data unless somebody had grown the crystals. You had to have somebody who understood how the X-ray equipment worked. And so there was this, you know, once the cloning started, there were people doing that. And I just look at those old lab photographs and sort of, I can label, he was a person who did molecular dynamics, and that was the girl who was brilliant at growing crystals and so on. I hope those photographs go into the new building somewhere, acknowledgement. And Guy loved biochemistry, and he loved structures, and he loved sort of seeing how the hydrogen bonds between this bit and that bit made it all fold up nicely and so on. So he was interested in everything that was going on. Rod Hubbard and his students were expert in the computing and also in molecular dynamics, and that was another aspect that made the crystallography more exciting if you began to understand how that happened. Tony Wilkinson and Jim Brannigan came and took over the cloning and that work and led that in the lab. Kevin Carton was interested in structure determination, and then there were lots of people who were solving interesting structures. There's Merrick and Fred and Gideon and Jean and, you know, all these people. The results were coming out, and it was lab talks were great, weren't they? And the other thing I'd really like to thank was we had this wonderful administrator, Caroline Myers, and she seemed, as I said, right down, not only to keep all the balls in the air, but she knew why they were in the air. So it was a tremendous help, and a lot of the lab success really does rest on somebody who understands what's going on. And it has been a great privilege for me to be part of that. And, you know, as I said, I was so lucky to wander into structural biology without knowing what I was doing. But that's another story. And anyway, now this new building that houses the equipment that's going to be needed to keep things going with the latest development. And it's funny because Guy just loved what they were doing with electron microscopy in Mill Hill, and he came back saying, you wouldn't know what Peter Rosenthal can see with his microscope. So to have it going on in York would have been so wonderful for him. And I'd just like to thank the people. I know it's been a huge struggle to get it up and running, not helped by COVID, but Johan and Jamie and many others whom you've thanked have had a lot of sleepless nights worrying about what was going to happen and where everything should be done. And I do hope that now the results are coming out, that structural biology in York will have an important role in the 21st century as well as in the 20th. So just thank you all for being here, and thank you all for, it's a wonderful discipline. And I must say it keeps you humble, because even if I knew what reciprocal lattice was, I never knew what PCR stood for.
Thank you very much for those words, Eleanor. I'm all the more delighted, having heard them, that we have been able to reflect the impact of the work that you and Guy have done and set in motion, and that will be carried forward further in the way that you did. So thank you again. And that finishes the evening. Thank you, everybody.